Bilirubin is a yellow pigment formed when the body breaks down old red blood cells, specifically from the heme part of the hemoglobin. It is a normal waste product, but because of its toxic and fat-soluble nature, the body must process it in the liver to make it water-soluble and excrete it safely in the bile and stool. Before we dive deeper into bilirubin metabolism, let's first understand the anatomy of the liver and the flow of blood and bile within it. This image shows an overview of the liver. The liver contains the portal triad, which includes the hepatic artery, the portal vein, and the common bile duct. The hepatic artery brings oxygenated blood. The portal vein carries nutrient-rich blood from the intestines, and the bile duct carries bile towards the duodenum. Here we can see a single hepatic lobule, and as you know, the entire liver is made up of many such lobules. Each lobule is roughly hexagonal in shape and is surrounded by 6 branches of the portal triad. Inside, we also see liver zones, zone 1, 2, and 3, where hepatocytes perform slightly different metabolic functions. The blood flow begins from the aorta, then moves through the celiac trunk, the common hepatic artery, and finally the hepatic artery proper, which supplies the branches of the portal triad. From there, blood enters zone 1, then zone 2, and finally zone 3, before draining into the central vein. The central veins merge into the hepatic veins, which empty into the inferior vena cava, returning blood to the heart. Meanwhile, the portal vein brings blood from the intestines carrying absorbed nutrients. This blood also flows through zones 1, 2, and 3 before entering the systemic circulation. So the portal and arterial blood flow in the same direction. In contrast, bile flows in the opposite direction. It is produced by hepatocytes, drains into bile canaliculi, and then flows outward towards the bile ducts, eventually leaving through the common bile duct and being stored in the gallbladder until needed for digestion. Bilirubin is synthesized at 3 primary sites in the body: the spleen, liver, and bone marrow. The spleen is the major site, where macrophages phagocytose old and damaged red blood cells to produce bilirubin. The second important site is the liver, where Kupffer cells, the resident macrophages of the liver sinusoids, break down circulating red blood cells. The third site is the bone marrow, where bilirubin is produced due to the breakdown of defective red blood cells resulting from ineffective erythropoiesis. Now, once the red blood cells are broken down, their hemoglobin is released and further degraded into 2 parts: heme and globin. Inside the macrophages, there's an enzyme called heme oxygenase. This enzyme opens up the heme ring and, in the process, releases iron and carbon monoxide while forming a green-colored pigment known as biliverdin. Next, another enzyme, biliverdin reductase, quickly converts biliverdin into unconjugated bilirubin. At the same time, the globin portion of hemoglobin is broken down into amino acids which are reused by the body. Now, this unconjugated bilirubin is not water-soluble. It's highly hydrophobic. So to move safely through the bloodstream, it binds tightly to albumin, forming the unconjugated bilirubin-albumin complex. From the spleen, this complex travels through the splenic vein, which then joins the portal vein, carrying bilirubin directly to the liver. From other sites like the bone marrow, it first goes through the systemic veins to reach the heart, and from there, it's carried to the liver via the hepatic artery. But remember, the majority of bilirubin reaches the liver through the portal circulation. Now, once the unconjugated bilirubin-albumin complex reaches the liver, it travels through the liver sinusoids, At the sinusoidal membrane of the hepatocytes, the unconjugated bilirubin dissociates from albumin and is then actively transported into the hepatocyte by a special carrier protein known as the organic anion transporting polypeptide. Inside the hepatocyte, an important enzyme called UDP glucuronil transferase attaches one or two molecules of glucuronic acid to the unconjugated bilirubin. This process is known as conjugation, and it converts the lipid-soluble or hydrophobic bilirubin into conjugated bilirubin, which is water-soluble. That means it can now dissolve easily in bile and be excreted. Once formed, the conjugated bilirubin is actively secreted from the hepatocyte into tiny channels called bile canaliculi. From there, it flows through the biliary tree, passing into the right and left hepatic ducts, which then merge to form the common hepatic duct. The bile can be stored in the gallbladder or directly flow onward. When we eat, especially a fatty meal, the gallbladder contracts and releases bile through the cystic duct, which joins the common hepatic duct to form the common bile duct. Finally, this bile, containing conjugated bilirubin, is released into the duodenum, where it helps in digestion, and further bilirubin metabolism continues in the intestines. When conjugated bilirubin reaches the intestine, particularly in the terminal ileum and colon, it encounters gut bacteria. These bacteria act on the conjugated bilirubin and convert it into a colorless compound called urobilinogen. Most of this urobilinogen gets oxidized to form stercobilin, which gives stool its characteristic brown color and is excreted in the feces. However, a small portion, roughly 10 to 20% of the urobilinogen, is reabsorbed back into the portal circulation, and this process is called the enterohepatic circulation. Now, when this reabsorbed urobilinogen returns to the liver, most of it is recycled and secreted again into the bile. But a tiny fraction escapes the liver's uptake and enters the systemic circulation. This portion is then filtered by the kidneys and excreted in urine as urobilin, which gives urine its yellow color. So, to summarize the entire process, when the red blood cell is broken down by macrophages, it releases heme. This heme is then converted into biliverdin, which is quickly reduced to unconjugated bilirubin. Now, this unconjugated bilirubin binds tightly to albumin in the blood. This bilirubin-albumin complex travels to the liver, where hepatocytes convert it into conjugated bilirubin through the process of conjugation. The conjugated bilirubin is secreted into bile and stored temporarily in the gallbladder. During digestion, especially after a meal, the gallbladder releases this bile into the intestine. In the gut, bacterials convert it into urobilinogen. Most of this is further oxidized to stercobilin, which gives feces its characteristic brown color. A small fraction of it is reabsorbed and returns to the liver through the enterohepatic circulation. But a tiny amount escapes and is excreted by the kidneys as urobilin, giving urine its yellow color. Now that we've understood the normal physiology of bilirubin synthesis and metabolism, let's look at some of the applied aspects of this pathway. Heme oxygenase converts heme into biliverdin, and it's upregulated during oxidative stress, like in sepsis, trauma, or hypoxia. But why? Because during severe stress, many cells and RBCs die, releasing free heme, which is toxic and promotes reactive oxygen species formation. These reactive oxygen species damage membranes, proteins, and DNA, worsening inflammation and injury. So the upregulation of heme oxygenase is the body's protective response. It breaks down toxic heme into biliverdin and then bilirubin. At moderate levels, bilirubin acts as a natural antioxidant, helping to reduce oxidative stress. Additionally, this process produces carbon monoxide and ferritin. Ferritin binds free iron released from heme breakdown, preventing further oxidative damage. Low levels of carbon monoxide, on the other hand, act as a vasodilator, anti-inflammatory, and anti-apoptotic agent. Important during sepsis, where blood flow and cell survival are crucial. However, in severe stress like septic shock, this protective mechanism can overshoot. Excessive heme breakdown leads to high bilirubin levels, overwhelming the liver and causing jaundice of sepsis due to increased level of unconjugated bilirubin in blood. Also, excess iron release may exceed ferritin's capacity, causing iron overload and further oxidative damage. So while heme oxygenase upregulation starts as a protective response, in severe illness it can become pathological, leading to jaundice and iron toxicity. And now the carbon monoxide production measurable in exhaled breath correlate with hemolysis rate. When heme is broken down, one of its byproduct is carbon monoxide. So every molecule of heme breakdown produces one molecule of carbon monoxide. As carbon monoxide diffused into bloodstream, where it binds to hemoglobin, forming a small amount of carboxyhemoglobin. From here it carries to the lung and exhaled in breath. So a tiny fraction of exhaled air normally contain carbon monoxide, produced by our own metabolism. So in hemolytic anemia, there is increased destruction of RBCs, result in increased heme release, and consequently, there is increased production of carbon monoxide. So exhaled carbon monoxide level directly correlate with the degree of hemolysis. More the hemolysis, the more the production of carbon monoxide. Drugs, for example sulfonamides, ceftriaxone, aspirin, can displace bilirubin from albumin. As they compete with bilirubin for the same albumin binding site, result in unconjugated bilirubin level rise in plasma. As this unconjugated bilirubin is a lipid soluble, so it can easily cross the immature blood-brain barrier of the neonates, causing kernicterus. So indirect bilirubin is increased due to displacement of albumin. But direct bilirubin, that is conjugate, is normal, as conjugation process is intact, and also rest of the LFT is normal. In some diseases, albumin level fall. For example, in case of severe malnutrition due to protein deficiency. In case of nephrotic syndrome due to albumin loss in urine. And in case of liver failure because of decreased albumin synthesis. As the result of decreased albumin level, there is increased level of unconjugated bilirubin in the blood. So in newborn, this can lead to kernicterus, and in adult, it can worsen jaundice and tissue deposition. Rifampin, which is the anti-tubercular drug, it inhibits the hepatic uptake transporter, mainly the organic anion transporting polypeptide 1b1 and 1b3 on the sinusoidal membrane. Now, unconjugated bilirubin can't enter the hepatocyte for conjugation. As the result of this, unconjugated bilirubin accumulate in plasma, result in jaundice. Now, this is the perfect time to understand an important mechanism of action of a drug called cholestyramine. Liver make bile acid from cholesterol. About 95% of the bile acids are reabsorbed in the terminal ileum and returned to the liver through the portal vein. This recycling is called the enterohepatic circulation. The cholestyramine is bile acid binding resin. It stay in the gut and bind to the bile acid. The bile acid cholestyramine complex can't be reabsorbed. It is excreted in feces. It interrupts the enterohepatic circulation. So bile acid pool in the liver is decreased. The liver must use more cholesterol to make new bile acid. Therefore, liver cholesterol level drop and the liver increase LDL receptor expression to pull more cholesterol from the bloodstream. Result: decrease plasma LDL cholesterol level. Therefore, cholestyramine is used as cholesterol-lowering drug. Because bile acids are bound and not reabsorb, fat digestion and absorption of fat-soluble vitamins can decreased. Stool may become bulkier and greasy.